Please join me in welcoming Catie Chang, our next presenter.
So yeah, thanks so much for the kind invitation to be here. And it's also really nice to have a chance to come back to NIH, where I was just recently a research fellow. And a lot of the work I'm going to discuss today was actually done in collaboration with many people on this campus. And so it's nice to have a chance to come back and also discuss that. So the topic that was suggested to me was on discussing how gathering more data in resting state is potentially more informative. And so here I'm going to be focusing mainly on intrinsic brain activity. So by intrinsic or spontaneous brain activity, I mean these natural ongoing signal changes that happen even in the absence of external stimulation or input. And in fact, these signals tend to be most often studied while we have our subjects just resting passively in the scanner. And what's been incredibly interesting, of course, is that even though it may just look like this is random to the eye, there's actually highly organized structure in terms of the correlations between different regions, also network patterns that tend to really closely align with known functional networks. And so this has sparked a lot of interest since it suggests we may be able to collect really important data on individuals even when they're just resting passively in the scanner. So as you can imagine, that's presented valuable opportunities to study individuals or patient groups who may not be able to perform tasks, maybe very small children or patients who may not be able to attend to stimuli or make behavioral responses. And indeed, there's growing evidence suggesting this activity could be a promising marker of both normal brain states and brain disorders. So for example, it's been shown that long-range correlations may differ across sleep and wake states found by Silvina Horowitz and Jeff Dunn. Also, Sue Whitfield-Gabrielli, speaking tomorrow, has found really interesting differences in connectivity modulated as a function of psychopathology and schizophrenia. There's also, Vicki Morgan has shown interesting signatures of a functional connectivity in epilepsy patients that correlate with seizure outcomes following surgery. And recently, there's been work showing that patients with depression may potentially be clustered into different subtypes based on their resting state patterns. There's also recent discussion and work showing that maybe it's more of a gradual continuum than clusters. So I think this is there's a lot of discussion going on here. But this is all moving toward the goal of trying to identify more biologically driven markers of depression and other disorders, which can perhaps improve upon standard diagnostic criteria or help with guiding treatment. And so, therefore, an important direction in this field for many years now is to see how we can try to maximize the information that we can derive from spontaneous or resting state fMRI signals, so that we can better use them to learn more about brain function, or about individual differences, or develop more robust indicators of neurological or psychiatric disorders. And so, towards this goal, one direction that we and others have been looking into is in how we can get more information from the temporal properties of this data. So much of the research has focused on the spatial properties or the spatial aspects of how these intrinsic signals are correlating. Sorry, my mouse is on. But we can also try to break this down into more time-resolved changes that may be going on. So not just in terms of connectivity, but also just any properties of the raw signals that may be changing over time. And the idea is that perhaps these temporal properties can convey richer information about brain function, about network dynamics, or about natural state changes like attention and alertness. So in this direction, there's been a number of different approaches for trying to look at more dynamic structure in this data. So for example, we might ask if there are different configurations of activity that the brain traverses over time, and if the distribution of time that a person spends in these different states might be linked with particular conditions like schizophrenia, which is work from Vince Calhoun's group. And instead of just summarizing the relationship between two areas as just one correlation coefficient, can we try to open that more over time and over frequency as well, using a time frequency analysis of the fMRI data to try to visualize how networks may be coupling or decoupling over time and over different time scales. And so it's been a growing area of research to try to advance markers of healthy and diseased brain function from these more dynamic features. But so fMRI is really powerful in giving us this tool for monitoring whole brain dynamic changes, but we also recognize that there's limitations in that if we're just using fMRI alone. So in terms of being able to try to concretely link the signal changes that we observe with new knowledge about brain function. And so first of all, the fMRI signal, as we know, is a slower and hemodynamic measure, and its temporal properties are, trying to get them, okay. And it's, right, so its temporal resolution doesn't approach the many of the millisecond timescales of many neural processes. And if we're trying to map functional connectivity or areas that have correlated fluctuations, then how do we try to interpret that? So if we see correlation between two different nodes, is there actually some kind of interaction going on between them? Or could those two nodes be receiving some common input from another third region? Or perhaps there are many regions that are modulated in common by some systemic physiological responses relating to breathing or CO2, for example, that can affect blood oxygenation and blood flow in multiple brain regions. And also, the time course that we see at any given voxel could be a complicated mixture of many different kinds of underlying processes. So maybe one component of a voxel signal may relate to some kind of active cognitive processing, but maybe a person is also getting sleepier over time, so it's also being modulated by circuits that are involved in brain arousal. And so in terms of, so there's many different promising directions trying to address this gap, I guess. And one direction we just heard from Vince is trying to use more data-driven decomposition, thinking across different modalities and trying to determine relevant features of different aspects of brain function. And another that I'll focus on in this talk is to try to use multimodal imaging, so simultaneous recordings primarily, to try to illuminate some of these questions and advance the use of resting state measures. So next, I'm going to discuss studies that use fMRI together with electrophysiology to try to look at dynamic brain states and specifically arousal, and then discuss some work on combining fMRI with TMS and invasive modulation in non-human primates to try to probe causal influences between different brain areas. So as you've heard a bit already today, scalp EEG is non-invasive. It provides information that's complementary to fMRI in that it has faster temporal resolution. It provides an electrophysiological indicator of brain activity. And so combining EEG with fMRI has been a promising way of trying to probe how these spontaneous fMRI signals relate to neural activity or to state change. And so in the EEG, for example, we can see these fluctuations. We can see these fluctuations in the algorithm, as well as other frequency bands that can serve as an indicator of how alert the subject is. And in this study, we found that if we looked across the course of a resting state scan in a time-resolved way, we found that windows of time where the EEG would indicate that the person was more likely to be alert came with different patterns of fMRI functional connectivity compared to windows where the subject more likely was more drowsy. And actually, one of my motivations for doing this study in the first place relates back to the work on time-varying analysis, where we started to do these wavelet analyses and other sliding window-based measures, but we always have this question of how much of that is really neural and how much is just due to sampling variability, since we're have fewer time points when we're looking at time-resolved things. And so trying to combine that with other measures can help us to gain more confidence that there's maybe some real neural changes or physiological changes happening when we start to look at fMRI data on smaller timescales. And for vigilance or arousal in particular, there have been other prior studies, such as work from Tom Liu's group, that has pointed out that changes in alertness seem to come with notable changes in fMRI data. So one of the findings is that there tends to be more stronger global fluctuations, as well as an increase in fluctuation amplitude, as shown in here in this work from Jeff Dunn's lab. And we've recently confirmed this observation in our own simultaneous EEG and fMRI data. So you can see here, the person probably started out more or awake, at least relaxed, so they have a lot of alpha power. And that the alpha then suddenly decreases, indicating the person may have fallen asleep, and you also start to see even more fluctuations in signal amplitude. And Laura Lewis had shown at ISMRM that also when you see a person waking up from sleep, you can see that amplitude attenuate again, and interesting dynamics happen in the thalamus. So if we think about alertness, I guess, maybe it's not that surprising that a considerable portion of spontaneous brain dynamics, or the fMRI signals we're looking at, may relate to changes in alertness. So it seems like the scanner, even though it's really loud, people tend to fall asleep. And this, this could be, we could view that as only just a confound in our studies. So if we were trying to do studies where we want someone to maintain a awake, alert fixation, then it's not good if some of them are going to sleep. But on the other hand, it also presents us a lot of opportunities into studying the brain, first of all, across different states of alertness, if that's what we're interested in. And also, and also trying to study perhaps how processes relating to vigilance regulation may be disrupted across different brain disorders. And in fact, because the fMRI signals and patterns do happen to change as with these changes in alertness, then we may be able to derive information about a person's moment-to-moment vigilance levels during the fMRI scan, and maybe without even the use of EEG, which is one of the goals we've been trying to move towards. And so in that, will give us more information from intrinsic signals that we can get maybe from a routine resting state scan. So along those lines, this was a demonstration of the power to try to be able to detect state changes directly from the fMRI data. So work by Taguchi. So this is so simultaneous EEG and fMRI were used here. EEG is the standard way of staging sleep. And here they found that if they take 1 or 2 minute windows of fMRI correlation patterns, that seemed to carry enough information to allow for classifying the sleep stage directly from the fMRI data alone, using a model that they, machine learning model that was trained based on simultaneous EEG and fMRI data. And of course, even before we fall asleep, we can still get substantial drifts in wakefulness. And I've shown this video a few times this year, and people always tell me that this makes them very sleepy to see this monkey falling asleep, but that's what's going on here. So this is a monkey in a resting state scan, so they're in a dark scanner bore. And apparently they find its resting state to be just as engaging as our human participants. So, they're just in the dark, and it's really a struggle for them to stay awake. I never get tired of seeing this. It's really interesting to me, actually. And this data were actually collected here at NIH by David Leopold's group, and they also had, at the same time, concurrent in electrophysiology and fMRI. And when we first started looking at this data together, I had started out with some different questions relating to networks and so forth, but once I started looking at these behavioral videos, it really highlighted how much we really can't treat the resting state as one uniform state, but there's these really strong modulations in arousal. And another thing that was interesting about this data set was that if you look at the fMRI data and try to find regions whose signals track along with these spontaneous eye opening and closure, which we might say is a really rough behavioral measure of alertness, then we found that most of the cortex seemed to be quite negatively correlated with these alertness changes. So you can see all around here, this blue indicates, blue indicates negative correlations, and this is more positive correlations that you find in more focal subcortical areas like thalamus. And so at first it was like, well, what to make of this pattern? So much negative. But going back to the literature, actually, this was not so surprising, because even the first simultaneous EEG fMRI study from Mark Cohen's group had shown that EEG alpha power fluctuation correlates with the fMRI data in quite a similar pattern. So then we said that, we thought that given that this pattern of fMRI signals that was correlated with this marker of arousal seemed to be quite reliable, we wondered if we could then try to go in the other direction. So if we only had fMRI data, if we learned this pattern somehow and then only had scans that had fMRI data, could we actually try to go, could we actually try to go in the opposite direction and be able to infer time courses of arousal fluctuation during an fMRI scan just from the fMRI data itself? So in practice, that could be really useful if, well, we know that a lot of big data sets, for example, don't have EEG or eye behavior recorded, and so it would be really interesting if we can decode that information from the data itself. And so in order to do this, we tried a simple approach where we just take this kind of a pattern and we call this a template pattern, and then we, this is learned actually from a set of simultaneous EEG and fMRI data. And then if you have a new unseen test data set, then you could do a sort of running spatial projection of that pattern onto each volume of that scan to try to estimate a frame by frame or TR by TR measure of alertness. And so this might be, for example, the time course of estimated vigilance across one of our scans. And to see if that actually may be working, this was a data set where we hadn't used yet the EEG or eye behavior that was recorded. So we actually used our template-based procedure to try to derive a time course of supposed changes in vigilance. And then we found that if we compare that with the actual behavior of the eye opening and closing that's going over time, again, this is in monkeys, then we could find it does a pretty good job of telling when the monkey had its eyes spontaneously opened or closed throughout the course of the scan. But then there's always this kind of question of when the eye behavior is opening and closing, how much of that difference is just due to that behavior versus a real underlying change in arousal, since we know that even when the eyes remain closed for a while, there's definitely, you can fall asleep. And so to make sure, to try to understand that, we isolated sections of the data that had long stretches where the monkey's eyes were closed, and we found that this approach did a pretty good job of estimating a measure that agreed with the electrophysiological indices of vigilance. So it might be more closely reflecting something like changes in brain arousal rather than just the eye opening and closing. And now we have evidence that this might work in humans too. So as here, for example, the fMRI-based estimate of vigilance agreed reasonably well with the EEG-based vigilance. This was in human subjects. And here we showed some preliminary evidence that we might be able to use this kinds of fMRI-based vigilance estimation to explain to some degree trial-to-trial variations in a person's behavioral responses, such as a reaction time in a working memory task. So we're trying to find better ways of doing this in our data, but the idea here is that basically if we can combine fMRI with other measures like EEG to try to figure out what are the kinds of signatures of these ongoing state changes in the fMRI data, then we might be able to then pull that information off of new data sets and enrich the amount of information we can get from resting state fMRI. Also, beyond just looking at alertness, there may be many other types of dynamic processes that we may be able to identify from fMRI data as well. People have looked at affective or cognitive processes, memory, for example. And in particular, Javier Gonzalez Castillo in Peter Banditin's group has shown that it may be possible to identify periods of discernible cognition that are ongoing during rest. And so along with alertness, we could imagine that if we can be able to focus in on these specific dimensions of the spontaneous activity, this might lead us to finding more targeted biomarkers of healthy or disease states. So, so far, a few messages are that, first of all, our fMRI signals are very rich. They reflect a complex mixture of underlying processes. And it's tempting to think that maybe this is just a confound. It is a lot of messy things in there. But we can really also view this as a lot of interesting information as well. It just means that we have to know how to identify the signatures of these various things. And once we can, then we might be able to use that knowledge to gain more understanding of brain function. So next I wanted to discuss a little how multimodal imaging can help us learn a little more about interactions between brain regions, specifically on how some of the correlated signal changes that we often see in resting states could perhaps originate or be regulated by input from some common source. And so in order to study that, we have to use tools that allow us to directly manipulate activity in particular regions and then we can combine that with fMRI whole brain to see what happens across all of the brain regions. So one technique for non-invasive brain stimulation is TMS. And so this was a study that we carried out several years ago with the Atkins group at Stanford, trying to see if we can use TMS to understand potential influences between major canonical resting state networks. And so resting state networks are typically mapped using correlation analysis in fMRI data, but from the fMRI data alone, it's not possible to tell whether there are interactions happening between different networks. But nonetheless, from many types of studies, there have been hypothesized to be interactions between networks. And so one is that the central executive network and salience networks, which are thought to be more cognitive or externally oriented networks, might downregulate activity in a default mode network, which is more of an internally oriented network. And so in order to test this, we define these individual stimulation targets by mapping networks using resting state fMRI, and then apply either excitatory or inhibitory TMS to nodes of these different networks with accessible targets and the medial frontal gyrus. And then what we found was that exciting these nodes did lead to more of an anti-correlation or negative relationship between those nodes and the default mode network. And that conversely, inhibitory TMS to those areas led to what seemed to be an apparent release of inhibition. So it changed some of the signal properties measured in the default mode network. And so, of course, there's so much we don't understand about this, and we're only able to probe a few nodes here. But I think the study suggests that there may be some coordination between these networks happening, and it proposes some methods that we could use to further examine that. So we know that BOLD fluctuations do show these very clear resting state network patterns. But in addition, we also almost always see this more spatially diffuse component having correlations over much of the brain. And this can often make up a really large fraction of our total fMRI signals. It's really common to refer to this as global signal, and in practice we usually mean the whole brain average. But it's also recognized that there are multiple sources that fold into what is that whole brain average, and which also might have slightly distinct spatial signatures. And this is all not yet completely understood, but more work going toward that direction of trying to understand that. So in terms of what contributes to these sort of global signals, on one hand, we may have certain artifacts like head motion that may change long distance coupling in some way. We also have physiological effects like changes in breathing volume over time, or heart rate or blood pressure, which can also induce large-scale changes in cerebral blood flow and blood volume. And when these systemic changes are affecting different regions in common, then it can act to increase their time series correlation. And just like EEG data can help us to parse out certain neural sources of fMRI, if we have concurrent measures of peripheral physiology, breathing, or pulse measures, or CO2, this could help us to understand and possibly differentiate physiological components and their respective dynamics. And as Molly mentioned, one way this is commonly used is to maybe clean up our data, but we might actually instead want to use this as valuable information to try to learn about brain physiology. For example, mapping the relative timing delays of breathing-related signal changes may help to reveal pathological changes in vasculature, or maybe perhaps help us to quantify non-neural aspects of hemodynamic response delays if we wanted to use fMRI to try to infer more things about neural timing differences. So it really is, yeah, I completely agree that one person's noise is another person's signal. And there's also evidence that sorry, I forgot to change the title, but there's evidence that some components of the fMRI global signals are linked with neural activity. So one key demonstration was the study from David Leopold's group in 2010, where they scanned monkeys during resting state fMRI, also with an electrode implanted in cortex. And they found that if you look at the high gamma band LFP recorded at a single electrode, if you convolve that with an HRF, then this ended up being highly correlated with fMRI data across the brain. So then we could maybe ask what kind of neural activity would have such broad correlations that we can see in the electrical and in the hemodynamics measures. And one candidate mechanism relates, as we've been discussing, maybe to changes in arousal or vigilance. And indeed, this paper, as well as the later study that I showed, in those studies, we were able to link these widespread fMRI signals with fluctuations in arousal that were measured using eye behavior, as well as lower frequency electrophysiological signals. And if that's the case, then it's known that many brain regions, including these subcortical areas, play an important role in regulating arousal states, and that they have long-range, widespread connections across the brain. And so we might hypothesize that these regions may be influencing some portion of the global cortical signals that we're seeing in fMRI data. And again, it's not possible to draw that conclusion only using fMRI data alone, because it's only correlational. But if we can specifically manipulate activity in any of these areas, then it may allow us to probe their role in causally influencing larger scale fMRI patterns. And so this study that I'll describe now is work with David Leopold's lab and Janita Turchi, where we had the opportunity to study nucleus spatialis, which is a region in basal forebrain that has many important functions relating to attention, cortical excitability, sleep-wake states. And here, Janita was able to transiently inactivate specific subregions in nucleus spatialis using mucimal injection, and then we performed long fMRI scans in order to see what happens to the spontaneous activity across the whole brain. And the main finding was that inactivating this region did seem to have very widespread effects on fMRI signals, and particularly for a signal component that was broadly shared across much of cortex. And so when we inactivate the nucleus spatialis here in one hemisphere, we find that the whole brain average signal correlations are really dominated by one hemisphere rather than coming more symmetrically from both of them, as we find in control data. And if we so we only inactivate one hemisphere per session, and so we then go and inactivate the same region but on the other hemisphere, then we can find that the directions, which is sides of the direction of the effect switched sides. And we can also see that if not only looking at these kinds of correlation maps, but also in the raw signals themselves. So if we go in and extract symmetric cortical ROIs here in temporal cortex, then you could see that the amplitude of these fluctuations was attenuated on the side that was ipsilateral to the injection rather than relative to the contralateral side. And that this also switched when we switched the direction or the side of the injection. But we also noticed that even though this is a kind of wide-scale effect, that this might have some degree of spatial specificity as well. And specifically, there are certain areas, such as the hippocampus and the cingulate cortex, that didn't seem to be disrupted as much in terms of what's happening on the ipsilateral side. So these kind of preserved their symmetry a bit more. And so when I first showed this to my colleagues who know much more about anatomy than I do, they found this really interesting because apparently the area in the basal forebrain that we inactivated for these experiments doesn't have anatomical projections to hippocampus and cingulate. And so it's known from literature that different parts of basal forebrain have fairly distinct long-range projections, and the effects that we started to see, there was indications basal would align well with the known anatomic patterns. So to go in and test that in more detail, we could then take another subregion of nucleus spatialis, this one called CH4AM. We could inactivate that as well and see if in the fMRI data there was a different pattern of areas across the cortex whose signals were impacted. And when we did find distinct patterns of areas that had that seemed to be affected in terms of their fMRI data, looking at this ipsilateral versus contralateral reduction, and that these specific areas did agree well with the anatomy literature. So areas that were reported to have weak or no projections from the area that was inactivated did seem to show a smaller effect in the fMRI data compared to those that were known to have stronger projections. And lastly, we asked what happens to the structure of resting state networks when we inactivate this region. So here are 6 resting state networks that I could see in the control data in monkeys. So there was no injection here, or there was injection of saline. And you could see that these networks, resting state networks in monkeys, are very similar to ones that we can identify in humans. And one possibility is that inactivating basal forebrain in one hemisphere could destroy the bilateral symmetry of these networks. Maybe it just becomes very lopsided. But another possibility is that we're really disrupting some more overriding common fluctuations and leaving the specific network signals more intact. And so in our data, the evidence seemed to lean more toward the latter, in that if we inactivate either the left or the right basal forebrain, then it didn't seem to really alter the pattern of networks very much. We could still identify them. They still had pretty symmetric amplitudes, except for when we do look more closely at the magnitudes, it was not the same. Actually, it was less on the ipsilateral side, but not quite as asymmetric as the global fluctuations. And so that still leaves the door open to the possibility that there is also some partial contribution to these networks. And then bringing this back to state changes, one thing that was clear from our data set was that it seemed the effects were very much dependent on what the arousal state of the monkey was. And so if I looked at sections of the data where the monkey seemed to be very alert, we either didn't see an effect, or interestingly, it almost started to lean in the opposite direction, where the contralateral side was less than the ipsilateral side. But when the monkeys started to become more drowsy and fall asleep, that's when we can see more of a disparity between the hemispheres and the signal amplitude, as well as these global correlations. And so one overall implication of this work is that disrupting this region did have very large effects in fMRI signals. And although this study was done in animals and was invasive, a possibility is that if it translates to humans, then this type of work may help us to learn what we might expect in human subjects if we see that brain areas are, or small brain areas like basal forebrain, are somehow disrupted. And so perhaps, if they're disrupted from degeneration, maybe in neurological disorders or through natural processes like aging. And there have been more studies along the lines of showing the importance of accounting for these neuromodulatory influences on fMRI signals and looking at the ascending arousal system. And for example, this was one study by my colleague Xiao Liu, who looked in the human connectome data and found interesting opposing dynamics between parts of nucleus procellis and a lot of cortical fMRI signals. We've also been looking more at autonomic or physiological correlates of arousal state change, since arousal fluctuations in different sleep stages also come with differences in heart rate, respiration, and other peripheral signals we can measure. And one thing that we noticed here came from looking at human overnight sleep data collected at NIH by Dante Piccioni. And one of the striking things that we noticed in looking at these signals was that in stage 2 sleep, whenever you get a K complex in the EEG, there seemed to be a very marked reduction in the amplitude of the fingertip blood volume signal. So this is a PPG signal, the normal kind that we always have on the person during scans. And so this is known to indicate things like vasoconstriction that might happen with a transient increase in sympathetic tone. So this relationship had been shown previously in the literature. And if we then look at fMRI data, we can see that these events seem to be accompanied by this whole brain spatiotemporal pattern that involved opposing signal changes between a lot of brain matter regions and areas in ventricles in the deep white matter. And so the particular mechanisms behind this response are described in this neuroimage paper here, and basically thought to arise from changes in the brain vascular tone that come with an increase in sympathetic nervous system activity. And generally, these kinds of findings might suggest that it's also really important to consider these autonomic effects and changes on the vascular tone in the brain when we're trying to study brain function. And now we're also looking at this kind of effect not only in sleep, but also in more awake resting state data. So to conclude here, multimodal studies can help us to gain more information about brain function and physiology from spontaneous activity. It can also help us to ground our interpretation of fMRI signals, for example, to link our observations to specific neural or physiological features, or to try to resolve directed interactions in fMRI networks, and therefore try to improve our use of fMRI in neuroscience and clinical applications. So I'd like to thank a bunch of my colleagues who at NIH who are involved in this work, and also big thanks to my lab at Vanderbilt. We're called the Nerdy Lab, and funding sources, and thank all of you for your time.